Hello everyone, welcome to this new series. Here we are going to post some lectures and conduct online classes especially for O levels and A level students. Soon we will start the other syllabuses as AQA and Excel as well. These classes and lectures will be posted by Dr. Rauf Ahmad, where he is a Cambridge certified teacher with a 20 years of experience.
हेलो आज का लेसन हमारा क्रेब साइकिल के बारे में। इसमें अभी तक हमने एसिटाइल कोएंजाइम ये जो लिंक रिएक्शन में बात करें उसको स्टडी किया, मतलब जो कि हम यहाँ पे देख सकते हैं। ये एसिटाइल कोएंजाइम ये बना है लिंक रिएक्शन में। तो लिंक रिएक्शन का मकसद ये होता है कि ये पायरुवेट को एक कंपाउंड एसिटाइल में चेंज करे ताकि वो क्रेब साइकिल के अंदर एंटर हो सके। एसिटाइल वो एक 2 कार्बन कंपाउंड है और पायरुवेट जो है वो 3 कार्बन कंपाउंड है। तो इसमें डीकार्बोक्सिलेशन हुई है। डीकार्बोक्सिलेशन में कार्बन डाइऑक्साइड गैस रिमूव होगी और उसके साथ ही रिड्यूस एनर्जी जो है वो फॉर्म होगा क्योंकि हार्डियन आयंस और इलेक्ट्रॉन यहाँ पे रिलीज हुए थे। तो लिंक रिएक्शन इस लिहाज से इम्पोर्टेंट है कि ये पायरुवेट को क्रेब साइकिल के अंदर रेस्पिरेशन की अगली स्टेज में एंटर कर रहा है और उसमें एसिडाइल फॉर्म करते हैं, जबकि कोएंजाइम इसको हेल्प आउट कर रहा होता है कि ये ऑक्सैलो एसिडेट के साथ इसको रिएक्ट करे, जो कि क्रेब साइकिल का फर्स्ट रिएक्शन होता है। तो इसमें हम ये चीज़ देख सकते हैं कि यहाँ पे जो ऑक्सैलो एसिडेट है ये एक 4 कार्बन कंपाउंड है जो कि मैट्रिक्स में होता है। लिंक रिएक्शन भी मैट्रिक्स में हो रहा होता है और क्रेब साइकिल भी मैट्रिक्स में हो रहा होता है। ऑक्सैलो एसिडेट जो है ये 2 कार्बन कंपाउंड एसिटाइल से कंबाइन करके एक 6 कार्बन कंपाउंड बना रहा है, यानी सिट्रेट या सिट्रिक एसिड भी इसको कह सकते हैं। जैसे पायरुवेट को हम पायरुविक एसिड कहते हैं, इसी तरह सिट्रेट को सिट्रिक एसिड कहते हैं। तो सिट्रिक एसिड जो है या सिट्रेट जो है ये फॉर्म होता है बाई कंबाइनिंग ऑक्सैलो एसिडेट विद एसिडाइल बॉन्ड लाइन। तो इसको हम फर्स्ट स्टेप के तौर पे लिख सकते हैं। अगर हम इसको डिस्क्रिप्शन में लिखें कि एसिडाइल को react with oxaloacetate to form citrate and coenzyme A release back. For the leak reaction, because coenzyme A do not go into the Krebs cycle. Only acetyl combined with oxaloacetate. So this is the first reaction of the Krebs cycle. Then in the second reaction, citrate on alpha ketoglutarate, and this is done by decarboxylation of citrate and dehydrogenation. Two reactions are going on the same path when citrate change into the alpha ketoglutarate. Here we can see alpha ketoglutarate is here, and it is formed by decarboxylation of isocitrate or citrate. You can say isocitrate is just a reacting stage, and citrate before it goes to alpha ketoglutarate, it changes to isocitrate. And then decarboxylation and dehydrogenation occur, during which carbon dioxide is released and anode is again which carry the hydrogen ions and electrons, which is being released from the citrate when it is going to be to the alpha ketoglutarate. So this is the second step now in the Krebs cycle, that citrate is decarboxylated and dehydrogenated at it form carbon dioxide, which is a waste, and hydrogen ions, which is accepted by hydrogen carrier, that is NAD. So it becomes reduced NAD. So now 2 steps are completed in the Krebs cycle. One step is that acetyl combined with oxaloacetate with the help of coenzyme A, and then the coenzyme A released back to the link reaction to carry both acetyl coenzyme A acetyl into the citric acid cycle. So what is oxaloacetate? Oxaloacetate actually is a 4 carbon compound which is always present in the matrix. So it is again available at the end of the Krebs cycle. So it is always there in the matrix of the mitochondria. The second step is that citrate is changing into alpha ketoglutarate. Here you can see alpha ketoglutarate is there, which is a 5-carbon compound because citrate is decarboxylated, so one carbon is removed. So carbon dioxide gets in the waste during the process, and then alpha ketoglutarate again decarboxylate and dehydrogenate to form succinyl. So succinyl is a 4-carbon compound because one more carbon is removed from here in the form of carbon dioxide, and the protons which are released during the reactions are carried by its carrier, that is again aniline, and one more aniline reduced aniline is formed. यहाँ पे ये चीज आपने ये भी याद रखनी है कि there are two Krebs cycles that are going on from one glucose molecules because there are two acetyl enzyme A over there, so two Krebs cycle, two citrate will be formed, and then two alpha ketoglutarate will be formed, and now two succinyl compound will be formed. So in each reactions of the first step, second step of citrate, when citrate change into the alpha ketoglutarate, two carbon dioxide gas will be released. So in one Krebs cycle, one carbon dioxide gas. Same like alpha ketoglutarate will change into succinyl. So again, one carbon dioxide gas is released. So the two Krebs cycles release two carbon dioxide gas. And here up till now, this one is the third reaction, and two reduced NAD will be formed in one Krebs cycle. Altogether, four reduced NAD will be formed up till the succinyl, which is 4-carbon compound. So again, I repeat 3 steps of the Krebs cycle. Number 1, this acetyl combined with oxaloacetate to form citrate, right? And coenzyme will be released back. Here you can see coenzyme is going back to the linked reaction because it is only helping the acetyl to combine with oxaloacetate. And then second step, in which citrate that is changing into the alpha ketoglutarate, which is 5-carbon compound during the decarboxylation reaction, and CO2 is removed back. You can mention isocitrate here, but it is not necessary to mention here. So you can write directly citrate is changing into a 5-carbon compound by decarboxylation, and a reduced energy is formed here. And then the third step is alpha ketoglutarate is changing into succinyl. Again, decarboxylation is occurred during the process, and reduced energy is formed. The next reaction, fourth reaction, is very important, in which succinyl is changing into succinate. So succinate is also 4-carbon compound. So there is no decarboxylation this time. But what is happening here, that ADP is changing into ATP. So ADP combined with phosphate in the matrix of the mitochondria, when succinyl is changing into succinate. A 4-carbon compound remain 4-carbon compound, but ADP is formed. So a substrate is changing into another substrate, during which ATP is formed. So this is substrate level या substrate link phosphorylation. तो इससे पहले जो हमने जिक्र किया था ग्लाइकोलाइसिस में, वहाँ पे भी सबस्ट्रेट लेवल फॉस्फोरिलेशन हुई थी जब ट्राइओज जो है पायरुवेट में कन्वर्ट होता है. तो substrate level phosphorylation you have when one substrate is changing into another substrate, and ATP is formed during the reaction by combining ATP with phosphate. So that is the substrate level SLP. You can say that SLP. So SLP, substrate level, substrate link phosphorylation. So this thing is to be remembered, that where ATP is formed during a Krebs cycle. So you have to remember when succinyl is changing into the succinate. This is the fourth major step of the Krebs cycle, and during which ATP is formed during a substrate level phosphorylation. The fifth step, again, you can see this succinate is changing into fumarate during a dehydrogenation process. There is no decarboxylation. So dehydrogenation means removal of protons and electrons. So this time the carrier is FAD, and it becomes reduced FAD. This is important, yet that you have to identify or locate where reduced FAD is formed when succinate is changing into fumarate. This is essential part of the exam questions also that you have to identify or locate which compound is found in which step in the Kreb cycle. So labeling of the Kreb cycles or identifications of the reactions can be in the exam question. So this is the fifth step that succinate is changing into favorite. There is no decarboxylation, but there is dehydrogenation. No doubt, it is enzymatic reactions. All the enzymes for each step is necessary in the link reactions and in each step of the Krebs cycle. So there are a lot of enzymatic reactions that are going on in the matrix of the mitochondria. So here, fumarate is now formed, which is a four-carbon compound during the dehydrogenation process when succinate changing into fumarate, and reduce FADs also. So this is in one Krebs cycle. Remember that there is always two Krebs cycle in one glucose molecules respiration process. So there will be two reduced FAD. So up till now, as a whole, if we can look at there is two reduced NAD and one reduced FAD. And if we count two Krebs cycles from one glucose molecules, then four reduced NAD and two reduced FAD. Right? So step number 6, when fumarate is changing into malate. So here it is not mentioned that water is added over here. So you can write here that water is being added in the reaction. Water is being added. So it is somewhat hydrolysis is going that fumarate is changing into malate. But it remains 4-carbon compound. From succinate to oxaloacetate, all compounds are 4-carbon. There is no decarboxylation. So whatsoever carbon dioxide gas is removing from the second step when the citrate is changing into the alpha ketoglutarate, and then when alpha ketoglutarate changing into succinate. So here is 2-carbon dioxide is formed on one Krebs cycle. Anyway, so we are here at malate now. Malate is formed and it is due to the addition of water here, and then malate is changing into the oxaloacetate, which is the end product you can see or compound in the matrix of the Krebs cycle. So oxaloacetate is reformed here and available for the acetyl, which is coming from the link reaction. And during this reaction, when malate is changing into oxaloacetate, hydrogen dehydrogenation occurs and hydrogen ions are removed, and these protons are being carried by its carrier again here NAD, and it's become reduced NAD. So here is a picture now. We can see that reduced NAD is forming here at when citrate is changing into alpha ketoglutarate, and when alpha ketoglutarate is changing into succinate 2 times, and when malate is changing into oxaloacetate 3 times. So 1, 2, and 3. 3 reduced NAD is, and one time reduced FADs. So there are 2 types of carrier, NAD and FAD, and 3 NAD will be reduced here, and 1 FAD is reduced here. So in 2 Krebs cycles, there will be 6 reduced NAD and 2 reduced FAD. So we have to look at the Krebs cycle as a whole. We have to look for what is the production of the Krebs cycle. So number 1, oxaloacetate is formed, which is being used in the Krebs cycle again. But we are looking for the compounds which is going on into the next step of the respiration. So we can write here कौन से ऐसे प्रोडक्ट हैं जो कि अगले स्टेप में होंगे. Number 1, ATP is being produced here when succinyl is changing into this succinate. This is one thing which we can use on that is the product of respiration. So ATP, you know that it is an energy currency, and there are a few features why we say ATP is an energy currency, because you have learned it before. So what is second thing which is being produced here during Kreb cycle, which can be used, or you can say it is a product of the Kreb cycle or the respiration, that is 6 reduce energy from one glucose molecules we are talking about.